Welcome to our review on cloning. So we've already looked at asexual reproduction in this topic, and if you remember, it uses mitosis, it needs only one parent, and because there's no mixing of genetic information, everything we produce is an identical copy, or a clone. So 3 examples of plants that use this asexual reproduction then. Our strawberry plant produces these things called runners, and then we get these identical copies of our parent plant developing at the end of each of them. We've got our spider plants, and again they produce runners with new plantlets occurring at the end. And finally our potato plant produces a thing called a tuber, and what we'll then see is those tubers develop into genetically identical plants. Another way we can produce these clones from plants is by taking what's called a cutting. So we start off with our adult plant, and literally we just slice part of the stem off. We then take the plant and dip it into this substance called rooting powder, which is basically a lot of hormones which will stimulate root growth. Put it into some well-watered compost and cover it with a polythene bag. And then that plant's going to develop its roots and grow as a perfectly identical copy to the parent plant. Another process we can actually use to create clones is what's called a tissue culture. So what we do here is we take a small piece of tissue and place them on a sterile jelly or in a sterile liquid using what's called aseptic technique. Now this is really important because if we're not using an aseptic technique, we risk contaminating it with bacteria and viruses which could destroy our culture. We then keep those cells at a suitable temperature and light level, and then they will develop into a whole new plant, again genetically identical to the parent. What we'll find then is when we're thinking about these different processes, plants are actually easier to clone than animals because they still have these stem cells in their meristems, which allows obviously the stem cells to become any type of cell required. The most famous example of animal cloning was Dolly the sheep. So Dolly was what's called a nuclear transfer clone. And what we can see in the diagram on the right there is the process by which they actually did this. So we started off with 2 sheep. Sheep A is an adult female Finn Dorset sheep, and sheep B is an adult female Scottish blackface sheep. What they did with sheep A was they actually took a normal body cell from the udder. From that they took the nucleus out, and then they just kept that safe for a moment. In sheep B, they took an egg cell and again removed the nucleus. This time, they destroyed the nucleus. That's not what we want. We want the empty egg cell. Then, they put the nucleus from sheep A into the empty egg from sheep B. They then obviously started it dividing by applying a little electric shock to it, and what we ended up with then is a little embryo developing. That was then implanted into a surrogate mother, and when the actual sheep was born, even though the actual mother of the sheep was the Scottish blackface, that was one that was carrying it, then they gave birth to an exact copy of our sheep A, the Finn Dorset sheep. So one of the big questions then is, why do we clone animals? What's the point in doing it? One of the key reasons is that if we've got any genetically engineered characteristics, we guarantee they will be passed on if we clone the animal. Now, thinking about human cloning, then what we can actually do is we can take any spare 8-cell embryos and we can split them and allow them to develop into cloned embryos. Now, we can use stem cells from these for research. However, we're not allowed to let them grow into a whole human. So there's lots of laws around this that very strictly govern what we can and can't do in terms of human cloning. So bear in mind that even though there is the potential to clone a human, we're not allowed to do it. So last thing we really need to consider then is the benefits and issues around human cloning. If we think about the benefits first of all then, one key thing, if we've got an identical clone of ourselves, then if we ever had an organ that stopped working, we've got a perfect copy right there for us to have as a transplant. So if you ever needed a new heart, take it from the clone. If you've got an infertile parent, then they can still have children. And because we'd be able to replace any aging tissues and organs with perfect copies from our own clone, then we'd be able to extend life as well. in terms of the issues or the downsides around our human cloning, then, we'd actually be creating an entire human life purely as a tool for medicine, or just as an organ donor. Any of those clones we create would be an identical twin of the actual cell donor, so you would have an identical copy of you, which obviously leads to several problems. If you've got someone who is absolutely identical to you, just a bit younger, same DNA, same everything, then that means that if they go and commit a crime, you might get the blame for it. And finally, we're going to decrease genetic diversity. If we just clone individuals, then we're not having that sexual reproduction, we're not having that genetic diversity, which is, in the long run, what's led to evolution of all the different species we see around us today. So, because of these issues, then there are lots of laws that prevent human cloning in the world today. So even though there are some benefits, it's not something we're going to be seeing anytime soon.